Hi, my name is John Ahlert, and I'm with my colleague Max Rubin, and we're second-year medical students at Midwestern University Arizona College of Osteopathic Medicine, and we're here to talk about our research on diet and exercise treatments on a diabetic obese model.
So as we all know, diabetes and obesity are a huge problem in our country. The prevalence of obesity is 42.4%, and the prevalence of diabetes is 10.4%. Diabetes has been shown to have some disturbances to intestinal function, and one of the treatments that we're studying in our lab, and in this study in particular, is GENISTEIN, which is commonly found in soy products and has been shown to have some anti-inflammatory effects.
We are specifically looking to determine whether dietary GENISTEIN or moderate exercise or both, or removal of that high-fat, high-sugar diet would reverse the obese diabetic phenotype and benefit the intestinal function of our mice.
So all of our mice were 6 weeks old, they're all male. Our lean controls were fed standard chow and water for all 24 weeks of the study, and the remaining mice were fed high-fat, high-sugar diet for the first 12 weeks, and then put into different groups for the remaining 12 weeks. Those groups being high-fat, high-sugar diet, standard chow, high-fat, high-sugar diet plus GENISTEIN, high-fat, high-sugar diet plus exercise, or high-fat, high-sugar diet plus both GENISTEIN and exercise. The high-fat diet was consistent with typical Western diet with increased saturated fats and carbohydrates, and the sugar water had a similar sucrose and fructose content as a typical soda. The mice that received exercise training had daily treadmill running for 150 minutes per week.
So we took tissue extracts upon euthanasia. The jejunum samples were used immediately for oosing analysis. Other tissues, including the jejunum, were flash frozen, and the jejunum and liver samples were frozen in OCT for histological analysis. And you can look at our statistical analysis below.
Here are our findings in terms of body weights. Body weights were taken every week for all 24 weeks of the studies. And there's a graph there showing the tracking of body weights on the left and total weight gain in the graph on the right. There's a significant increase in the high-fat, high-sugar group as compared to the leans, the high-fat, high-sugar group being in black and the leans in white, shown in both graphs, as well as a reversal with the removal of high-fat sugar diet in the standard chow mice, shown here in gray, in both graphs, and a reversal, significant reversal in the GENISTEIN plus exercise group shown in blue.
We also took a look at liver weights measured in a liver weight to body weight ratio, and we found that high-fat, high-sugar diet significantly increased that ratio in the black, and that increase was reversed with return to the standard chow, also by the exercise, and also with the GENISTEIN plus exercise. And this is important as a potential treatment for non-alcoholic fatty liver disease.
To get a better understanding of that increase in liver weight that John was just talking about, we looked at a few markers of hepatic steatosis, being percent fat area, number of droplets, and average droplet area. All three of these measures were increased in the high-fat, high-sugar diet as compared to the leans, and significantly reversed with the standard chow group. Here are some histological images on the right showing fat deposition and reversal of that fat deposition. And this is important because it indicates that the increase in liver weight is likely due to fat deposition, and there can be some benefit to removing that high-fat, high-sugar diet.
We also took glucose tolerance tests at 12 and 24 weeks. And in the black, on the top graph there in the black, you can see the high-fat, high-sugar group having a higher glycemic level, basal glycemic level, and less of a return to normal glycemic levels. But in the gray circles there, with the standard group, you see a, quote, lean-like response and a more of a return to normal glycemic levels. GENISTEIN exercise and Gen X all offered a little bit of improvement, but we're still waiting on the statistics.
So we also utilized an oosing chamber, which is an ex vivo model that gives us a measurement of chloride secretory function in the intestine. And basically we just took samples of jejunum immediately after euthanasia, placed them in the oosing chamber, and were able to get a measurement of chloride secretion.
We found that that basal chloride secretion measured at 20 minutes was significantly increased in the high-fat, high-sugar diet there in the black dots there compared to the lean. And that decrease was reversed, aka increased with return to the standard chow in the gray there, and that's in both graphs. And this is consistent with our previous research that has shown chloride secretion is significantly decreased in a variety of murine diabetic models.
We also added bumetanide to the oosing chamber, and what bumetanide does is it inhibits the basolateral NKCC2 cotransporter, shown here in red. We found a similar trend in secretory function as what John just showed you on the previous graph, which indicates that the changes in secretory function are chloride channel mediated.
We We took a further look at the chloride secretion and found, specifically at CLC2, which just represents a family of luminal chloride channels. And basically that CLC2 was significantly decreased in the high-fat, high-sugar group, as you can see in the black. And there was a bit of a trend for reversal with the standard chow group in the gray.
We also looked at intestinal morphology via crypt depth, and what we found is there was no significant change and no real trend, which indicates that the changes in secretory function that we found were likely not due to morphological changes in the intestine of our mice.
We also took a look at senescence markers, specifically PH2AX, which is a marker for DNA damage. And we found that the high-fat, high-sugar diet significantly increased that expression there in the black, but that expression was reversed with the removal of the high-fat, high-sugar diet, the standard group, and then also with the genistein group and the genistein plus exercise group.
We also looked at serum inflammatory markers such as TNF-alpha, and we found that expression of TNF-alpha was significantly increased in the high-fat, high-sugar group as compared to the leans, and that increase was reversed with the standard chow group, the genistein group, and the genistein plus exercise group.
Another inflammatory marker we took a look at was IL-10, interleukin 10. We found that that high-fat, high-sugar diet significantly increased that IL-10 likely response to the obesity-induced inflammation, and that increase was significantly reversed this time by the exercise group there in the orange.
So in conclusion, the elimination of high-fat, high-sugar diet seemed to have the most beneficial effect out of all of our treatment groups. It resulted in maximum weight loss, maximum benefit to hepatic steatosis and return to normal glycemic levels, normalizes intestinal chloride secretory function, reverses intestinal DNA damage, and improves inflammatory state. Our data also suggests that genistein and/or genistein plus exercise may have a comparable role in inflammation, senescence, and hepatic steatosis. And this study definitely has some important implications in terms of the reversal of diabetic obesity, and future studies will be needed to examine the mechanisms of action.
We'd like to thank Midwestern for the opportunity to get this data, our lab mates, of course, and also to the ACP for the opportunity to present this research. Thank you very much.